Hello and welcome to the Genetics Podcast. I'm your host, Patrick Short. My background is in population genomics and studying the genetic causes of rare disease. I did my PhD at the Sanger Institute in the University of Cambridge and have been in biotech since 2018 when I started Sonogenetics. Sonogenetics helps academic and industry researchers to run large-scale genetic testing programs that speed up their clinical trials, generate data sets for the next big breakthrough, and give participants the best possible experience taking part in research. Each episode of the Genetics Podcast, we bring you insights from the leading minds in genetics and precision medicine, including household names and Nobel Prize winners, as well as early career scientists and biotechs working on the next big breakthrough. Whether you are a scientist, entrepreneur, executive, patient advocate, or simply someone curious about how genetics shapes our world, you're in the right place. Thank you for listening, and let's get started. Welcome, everyone, back to the Genetics Podcast. I'm very excited to be here today with Terry Pirovolakis, who's the founder and CEO of Elpida Therapeutics, which is a biotech that's on a mission to bring gene therapies to kids with ultra-rare disease. And he has an amazing story that starts with his own son and has blossomed into an incredibly impactful and inspiring biotech company. So, Terry, thank you for taking the time to be with us today.
Thank you for having me. Really appreciate it.
I would love to go from the beginning. When Michael, your son, was diagnosed with SPG50, and it'd be great if you could tell people about the disease and a little bit about your son as we work through this. But when he was diagnosed, what went through your mind at that time? And then how did you decide to take matters into your own hands and become, effectively a scientist CEO when that wasn't your background before? I'd love if you could just take us there.
Yeah, I mean, it was, I call it a day. We have PTSD from that day still. The second they told us that Michael had SPG50, that they knew very little, that they did know that he'd be paralyzed from the waist down by the age of 10, quadriplegic by the age of 20. He'll never walk, he'll never talk. We immediately went into a fog. We were crying. We were, like any parent was just, hysterical. And I said to them, get a room ready. I'm going to do a harikari myself, and we're going to just take anything out of me that we need to do to save Michael. And they said, Terry, unfortunately, that's not how genetic diseases work. We went home that night, and the way they do it is they send you an email a few hours later, and they tell you more about the disease. So we went home. We met an amazing family out of Boston who had a very similar disease, part of the complex. And they started talking to us about gene therapies and ASOs and drug repurposing. And it was like, way over our heads at the time. And an hour later, we got the email, and I just went down the rabbit hole and just spent 24 hours in the room straight researching everything I humanly could. And we realized that there was something that could be done. It's going to cost a significant amount of money and could take a decade. And that was kind of the precipice of our journey, which is, trying to save Michael. I came out of the room 24 hours later, or my wife came in and said, what are you doing in here? And I said, we're gonna have to make Michael a therapy. We're gonna have to raise $4 million. We're gonna have to sell our home, liquidate everything that we have. And are you okay with that? I'm gonna have to travel a lot and find these people that we're gonna have to build our team. And she said, just do whatever it takes,
Wow. When you set off on that journey, how did you arrive at that figure, and what was the the plan on the napkin that you had, and how did that play out in reality? Because you did raise a ton of money and you have pulled together a ton of experts. Maybe you could talk about what the initial plan was and then how you actually brought that into the world.
Yeah, so I started researching companies, institutions that were experts in the field. And the good thing was that I knew that if I attended a conference, I'd be able to meet these experts. Thankfully, a month after Michael was diagnosed, there was a conference called ASGCT, American Cell and Gene Therapy Conference. It was in Washington, I think it was either Washington or Boston. And so because of my background in IT and building these large infrastructures, I knew if I started probing and interviewing, not asking, interviewing people who I thought I wanted to be part of my team, that we could find the right team. So I went to ASGCT and I met 6 to 7 world experts in the field. I then went to Sheffield, England. I met the 7th. At the time, there was a lot more, but these are the people I was told to go see. And then I interviewed people at ASGCT and asked companies and institutions, who would you go with? And 90% of the people said Dr. Stephen Gray. So after doing my research and meeting all these individuals and doing my gut check, I knew he was the right person as well. And I sabotaged him on the stage at ASGCT after he won an award. And at the time, he was just really busy. He couldn't take on any more programs. And I said, you got to figure it out because we're going to work together. We're going to make this happen. And he was fully on board. And from then on, we became tied at the hip. And then, slowly our team grew to, an FDA expert, a toxicology expert, a CMC expert, and that became our core team. And then, we had amazing people like John Crowley and Emil Kakis, who were our mentors in this journey and guided us along the way.
What was the hardest thing for you to learn not coming from this background? Because it's a hugely interdisciplinary thing. You've got regulations, you've got the chemistry, you've got the biology. What was hardest for you, and how did you crack through that?
Well, a month and a half after Michael was diagnosed, I took a cell biology course at Sick Kids. Well, I bombed it. I basically just I attended without signing up and just listened to it, and I found that I was just going too slow. So I researched more and more and succeeded past that. And I think it was just taking things in chunks. You can't absorb everything. You can't be a manufacturing expert and understand
Yeah.
how everything goes. So what I did was I let Dr. Gray tell me, we're going to be doing this step, and then we're going to follow this step, and then we're going to follow that step. And as we did each step, I started researching and going deep diving into what that step was, because it really didn't matter what the next step was or two steps beyond that, because we're not there yet, right? The
Yeah.
the step that we were in mattered the most, and maybe the step afterwards. So that's why I focused my time really understanding, okay, well, we have to make a mouse. What does that mean? How do they make the mice? Who do they use? That kind of stuff, right? Or we have to make a research grade vector. What is that? How does that work? Who are the experts in the field in doing that? And so basically, I just every time we were doing things, I would probe Dr. Gray, bug him for a little bit, and then really understand what that meant to move forward.
Was there a time driver that you were on where you mentioned that the doctors told you Michael would lose the ability to walk by and be paralyzed from the waist down by 10, fully quadriplegic by age 20? Did you then therefore were you on a clock in a really real sense to say we have to get this done by X date? And how did that play out? Because that is uniquely stressful when it's your own son.
Yeah, like Dr. Gray, when we were initially, he was very respectful and really set the guidelines. As he told us, I don't do N of one. If we're going to do this, it's going to be for all the kids. So we were fully aligned. He said that, this takes, 4 to 5 years if you're lucky. It really depends on how well the mice work. There's a lot of factors that are there. But if we really worked hard and he goes, if you were able to raise the money to make things move faster and take significant risks, we could cut it down to 3 years or less.
Wow.
And for me, I thought, okay, we're going to make this in 18 months. That was my goal, is to take it in 18 months. Unfortunately, we had a lot of roadblocks, but COVID, which was a massive one. But I think, based on COVID and based on, our disease, having no mouse, having no cell lines, nothing, I think we did pretty good for being under 3 years, from zero to treatment.
I would say so. What did you learn? What did you learn in that process about how you could scale this out to other cases? But also if there is anywhere that you could have shaved some time off, that 3 years is very impressive. But I know when you look back on it, you had said, "Oh, we spent too much time on this aspect or that aspect, but not enough somewhere else."
I don't think we could have shaved anywhere, to be honest with you. I mean, there was obviously COVID, but I mean the Dr. Xin Chen and Dr. Stephen Gray just really took this seriously, like they do with all their projects. And they did everything one step after another, or in most cases concurrently, where they could. I mean, during the height of COVID, where most places were shutting down their cores, Dr. Xin Chen, who was the associate professor at the time, went in every day and made sure our mice were alive and continued our colony. And that was the biggest piece that we had to do because we had to show efficacy in the animals. And the longest lead time that we had, unfortunately, was to breed the animals, bring up a colony, dose the animals, and that's what the longest lead time was. But once that happened, we were able to do things concurrently. For example, we were doing our efficacy studies while we were doing our tox studies in rats.
Right.
We were doing our we were manufacturing the drug while we were doing the tox study. So we didn't skimp on safety in any way, shape, or form, but we did do things concurrently. And if that would have failed, we would have thrown away the $4.5 million that we raised, and not been able to serve again, basically.
Where did you raise the money from? How did you, was it a couple big donors, a big group of people? How did that shake out? I'm just genuinely curious.
Yeah, we thought, because I worked for a large bank at the time, and we were working with large companies like Salesforce and AWS, and I thought, oh my God, we're gonna rock this. We're gonna
Yeah.
raise $3 million in a couple of weeks, and it didn't work out like that at all. Our community, we came out to our community about 2 weeks later. I knew that I had to do a GoFundMe. I knew we had to make a video, so we paused and we did those before coming out to our community. I also wanted to make sure that we had a contract with UTSW and an agreement that we were going to move forward. Because if we just came out and said we're going to potentially start something, it's different than we started something. So we liquidated our life savings. We signed an agreement before we even started raising money. And then we came out to our community, and our community just rallied behind us. I'm in a small town, a small suburb of Toronto, a small microcosm. And then 2 weeks later, they did a beer works for Michael. They made a beer. They did an auction. We raised about $25,000. And then people just started calling us. I want to do a gala. I want to do a lemonade stand. I want to do, a bowling event. I want to do a soccer event. And then we did
Wow.
events too, like we did a golf tournaments, we did a gala, we did a bikeathon. But basically, it was 20,000 individual donations on GoFundMe and some very large events and one or two amazing large donors that got
Yeah.
us there.
It's incredible. And you now fast forward to today, so I think Michael was the first to receive the treatment in 2022. You've had five kids in total, and including Michael, that have received the gene therapy. Have I got those numbers right?
Kind of. So
Yeah.
Michael, so what happened was we were always intending to treat more children, but at the same time, we didn't want to be traveling around the world during COVID. So we were fortunate that enough that Sick Kids agreed to treat Michael at the hospital. So we applied to Health Canada December 10th. December 30th, we got a response back, and he got treated 3 months later, like you said, in March of 2022. And then we had two more doses available to us. So we opened up a phase 1/2 in Dallas, Texas, and we treated two children, one from the US, one from Spain. And then we were fortunate enough that the manufacturer made another batch of drug for us at no cost. This was Virogen in Spain. So we treated two more children in compassionate use in Spain, a 14 and a 17-year-old. And then we started treating babies under 2 years of age. So in total, we've treated 9 children so
Wow.
far. And we're opening up, as we speak, we're opening up a site in Denmark for two more children, a site in Bergamo, Italy for another five. We're opening up a site and we're opening up a phase 3 in Dallas, Texas, and in SJD Hospital in Barcelona. So in total, between this year and Q1 of next year, we hope to treat about 18 children more with this
Wow.
disease.
How is that going to scale, and how will that continue to scale? It sounds like you're getting a lot of support from the early partners who have helped. Is there a way, to partner this out so that somebody else can take it to as many kids as there are? I actually don't know the epidemiology of the disease. Like, how many kids are there out there that should be considering this?
Yeah, so the dilemma that we have is that it's non-commercially viable. I think the commercial viable number is around 5,000 children in the U.S. We're at 100 in the world, 13
Wow.
in the U.S. So it's ultra rare. What happened was when Michael was treated, I tried giving away the program. I basically said to companies, institutions, take it. Here's the drug, here's the license, here's everything. You can have it for free. I want nothing of it.
Right.
And everybody kept telling me, we're sorry, Terry, that this is not the way things work. It's ultra rare. We can't take this program on. So then we were able to, thanks to two very amazing philanthropists, move this program forward. Sheila McHale and Javier Garcia are the two people that really push us forward and allowed us to create Elpida Therapeutics in order for us to not only take on SPG 50, but take on other rare conditions that were either stuck in academia. That was the original goal, right? Let's get them stuck out of academia. But then when the biotech collapsed, basically over the next couple of years, a lot of programs were ready to go, either manufactured and ready or filed an IND, and we're stuck. And those are the programs that we took on. CMT4J was the second one, CLN7 the third, and we've taken on three more that are unfortunately we haven't licensed yet, so we don't talk about them. Because usually what happens is until we can fund them into a trial, we don't license them. We don't want to hold them up from anybody else potentially moving them
Yeah.
forward. And then so that was how we started with Elpida. We got those programs in, and then around December of this year, the Trump administration or the Congress denied the renewal of the peer review voucher. And what that led to is funding being completely gone, evaporation basically from the rare disease community within a single swift,
Yeah.
vote from one congressperson. And what happened was we were about to go bankrupt as a basically a nonprofit. And then we started trying to give away our programs to larger institutions now that we had data. You would think
Yeah.
you have data and a drug product. And again, no one wanted it. So we came to this precipice where we either had to change the fundamental way we're working or these programs would never see the light of day. So we opened up a consulting division of Elpida Therapeutics and started consulting for proof of concepts, IND
Yeah.
filings, pre-IND filings. And now our employees are paid for through this consulting division, and we can then focus in on grants and patient funding to fund the programs we're running.
Do you have any insights on how we solve for this commercial viability problem? Is it an issue on if the cost were to get to a certain point, then it might be viable because there are millions of kids, millions of adults as well that are trapped in this ultra rare category that collectively it's a lot of people, right? Depending on the estimates, could be 5% of the population even in these
It's one in
unaddressed.
10, really, that's what it is, right?
Yeah.
So, unfortunately, it's like monoclonal antibodies, right? This is we're in the same kind of predicament where they were really high at a certain point and they came down based on more of them coming out. And I think until we have about 30 to 50 gene therapies that bring down the cost, that bring down the manufacturing, that allow us to understand it better, to allow yields to get better in manufacturing, insurers to pay for more programs. Unfortunately, that's when things are going to happen. With eight gene therapies approved right now, and the bar being so significantly high, going from a clinical grade product of, and I think the main problem is manufacturing.
Yeah.
Your drug is either going to show efficacy or not show efficacy, and it's really comes down to having good endpoints, which we don't have as a community. We don't have really good endpoints, but if you're lucky enough to have a biomarker, then you can move things forward. But even if you got past your endpoints, going from a clinical grade product to a commercial grade product isn't like walking up 10 steps. It's like climbing Everest,
Right.
right? It's a significantly different a goal to achieve. And I think what we need to do is we need to work with the regulatory bodies to say this bar is okay for a disease that has 100,000, a million people in the world, but for 13 in the US, this doesn't make sense.
Yeah.
You
What are
know,
the kinds of differences between the research grade and the commercial grade that,
it's all about validation,
yeah,
right? Characterization and validation. So, I was told a very simple analogy. Let's say that you have to meet a target of pH, and you have to go from, your range is from 5 to 6.
yeah.
You may have to do 30, 50-liter runs to validate your pH, you know,
Wow.
and those cost money, and
Yeah. And that's just one parameter, right?
that's
You
one
got
parameter. I may be
dozens
over
of others.
exaggerating, but that's
Yeah.
I mean, so even if it was 3 50-liter batches, it's still a lot of money, you know.
Yeah.
So that's the problem. Or if you have to do 1,000-liter run, as an example, what does that look like? And that might have to be done for certain aspects of it. So that's the dilemma, that you don't have to do those things for non clinical products, but you do for commercial products.
Yeah. So you basically have this huge fixed cost in the form of not just manufacturing the actual product, but all of the associated validation and regulation that you have to overcome. And then if the number you're dividing it by is 100 patients worldwide, then it's just impossible to make the
Yeah,
numbers.
and people say, why don't you just even open IND? It sounds simple, but let's put it this way: if it costs you somewhere in the neighborhood of $400,000 to $500,000 to make the drug, and then it costs you in a U.S. hospital another 300,000 to 400,000 to treat the patient, and then
Yeah.
you know, we don't use CROs, but let's say you brought in a CRO and you had to pay another $500,000. So then your minimum cost to run that trial is around $1.5 million. Which family can afford that? You
Yeah.
know, you might be able to find one or two families that have extreme wealth, but what about the families that are working in a factory or, they are middle income, and what are they going to do? They're going to sell their home and everything they have. It's an unviable process. So
Yeah.
the only way for us to get this into every patient is to get it through, get it approved, so insurers and governments pay for it. But the other benefit is, which I think is even bigger, is it then gets on the newborn screening panel. So instead of us treating 5 month olds, 1 year olds, 17-year olds, we're going to be treating the babies at 2 months old, 1 month old, and they're going to be, more towards the cure than they are towards the treatment. I call it a
Yeah.
sliding scale. You catch a kid at a baby at 1 month, 5 months. That's more. It may not be a cure. It might be more towards a cure. And then you slide that scale to where we got Michael, at 4 years of age, it was more towards a treatment. And if you slide it out to a 20-year-old that is already quadriplegic, that's less of a treatment for that child. It may be not even a treatment. It may be
Yeah.
something where the risk ratio doesn't work anymore, right?
Yeah. I know you've been seeing the same news that I have about gene therapies more broadly. My interpretation of it is as more and more gene therapies are moving into clinical trials, and also as they move into different populations, we're starting to see more safety issues come up primarily, and that's what's catching the news. But what I don't know is that's just proportional to the scale. We're treating more and more people. There are more and more trials coming online. I'd love to have your perspective on what we're learning right now about the viability and future of gene therapies. Is this an expected moment in time as the technology continues to mature, or is it teaching us something about this is maybe going to be harder than we think to get to the scale that you talked about we need 80 or 100 approved therapies to start to get those economies of scale?
Yeah, I think no matter what trial you do, you're gonna see AEs and SAEs, right? It's inevitable. I mean, you're tapping into someone's CSF, you're injecting a product. They might have immune response, they may not. They may be crim negative and never seen the protein. They may be crim positive. You don't know these things, right? And I think that's where, Jim Wilson, when he first did that gene therapy and killed the poor child, and people didn't realize there's an immune response to this gene therapy. And then we realized that, and now we have regimens, but they're not the greatest out there. We don't know when
Yeah.
to take patients off of them. And so I think there's these things are learning curves, and it's an unfortunate thing that we're gonna have patients that are gonna get sick and potentially die. Um, but in these fatal conditions, what other hope do these children have? And I think we have to figure that out and make sure that we're doing a risk reward profile. For example, maybe we don't treat the 17 or 20-year olds, right, that are already quadriplegic,
Yes.
right, and put them on a lifelong immune regimen. But maybe we catch them at the latest when they're still mobile, but they still able to use their hands, so they can feed themselves. And maybe that's the limiting stopgap. I don't know. That the problem really
Yeah.
comes down to we just don't know enough. Not enough data is being shared around what's going on with these SAEs. Everybody's
Yeah.
keeping them close to the pocket. And I think until we have this mechanism where we can openly share them without being um, discredited or penalized, I think we're gonna be limiting ourselves. I think what has to happen is if you do a compassionate use for a child that is very severe, and the family really wants to have this treatment done because their child's going to die or whatever it may be, that the FDA shouldn't penalize us for that treatment, right? If we're going outside of our comfort zone, but there's still a risk reward ratio there for that child, then we shouldn't be penalized and then have our drugs stopped or potentially
Yeah.
completely removed. I know it sounds crazy, it's difficult, but that's the reality of things if we're gonna treat the children that exist today. If
Yeah.
we, if we forget all of the kids that exist today, then the ratio goes down significantly. But if we want to give the children that are living today a chance, then the risk profile is going to go higher.
Yeah.
That's the reality of where we're at right now.
Yeah, and I think the principle you outline is a totally reasonable one. When you're looking at your pipeline of therapies you're developing at Elpida, but also the clients that you consult for, how are you thinking about the plethora of different precision medicine options that there are beyond viral vectors, gene therapy, but we also have gene editing, we have RNA. I don't know in the case of SPG50, for example, whether any of these other modalities might have been viable route. But today, when you're starting a program from scratch, you have probably more options than you've ever had to think about small molecules, gene therapy, biologics, depending on the mechanism that you're going for. How do you go about navigating that maze, either yourselves or with the clients that you work with?
Yeah, so for myself, SPG50, when I went down for looking for Michael's therapy, I went down the rabbit hole and looked up LNPs and other vector options because, I wanted to make sure we could maybe start off small and then we'll work our way up and not have the problems around AAV manufacturing and dosing. But unfortunately, before COVID, there really wasn't anything with around LNPs that was really moving forward, at least for the brain. Everywhere else for me a bit, but not for the brain. And then, so AAV was really the only option. ASOs wouldn't work for Michael because he has a nonsense mutation. So you can't upregulate anything because nothing exists to upregulate.
Yeah.
And then we started looking at small molecules and things to the NIH under a very specific group. They ran a drug screen for about 500,000 small molecules, and the problem is it's a very, very small chance that you're going to find a drug that one is safe, two that's going to pass the blood-brain barrier, and three that is safe enough in the volume or in the dose that you need to again pass through the blood-brain barrier and not harm your child.
Yeah.
So we found a few, but none of them were worth pursuing. So every program that we take on and support, we look at all the options, but we are moving away from viral vectors like AAV9 specifically, and looking at other vector options. We're also looking at gene editing, but we usually look for gene replacement therapy first because here's an example. Let's say that you have a population that has 20 variants, and this child has a variant that might affect 2 of 1,000 people,
Mm-hmm.
right? But if it's small enough and if it's into a gene therapy, then you can affect the entire population. So what we look at is, okay, we say, okay, is this variant across all the population or a majority? Is the gene too big to fit to a regular vector? And then we have to do gene editing. Those are the things we look at before we decide what if we're going to be doing gene replacement therapy or gene editing. But right now, it's all the programs that we have in our pipeline that are not consulting are all AAV because they're 3 years ago programs, right? So they're all AAV based.
What are you shifting towards, LMP or, and just for everybody, AAV is adeno-associated virus. It's one of the most common, probably alongside lentivirus vectors for getting the gene into the cell so it can do its thing. But you can also wrap the gene up in lipid nanoparticles, LMP, and there's other.
These are receptor vectors that are being created, right? There's a whole bunch of stuff that's being done. So we're
Yep.
based on program by program, size of the gene, where it needs to go. We look at each one individually. You have to hit oligodendrocytes. You have to hit neurons. Is it a liver mediated disorder? What are you trying to hit? Is there a problem with the liver in this disorder, and we have to or kidney that we have to avoid? All these things come into play while we're looking at what options there are.
Yeah.
But in most cases, it's very complicated, right? And so we look at them individually, one by one.
Yeah. When in clinical trials, my understanding is that you all have often approached this with less of a rigid phased approach and more of a convertible or continuous enrollment type approach. I'd love to just hear a little bit more about that, and also how common is that, or how common is that becoming in rare and ultra rare disease to take something that's a little bit more agile and adaptive versus what everybody sees in the textbook, which is rarely the case, right? Phase 1, phase 2, phase 3. You have to throw that playbook out a little bit for rare disease.
Yeah, I think I think when you do a phase 1, 2, and 3, that's usually for larger indications, and the purpose of that is to really narrow down your endpoints, right? Make sure that you have the right endpoint, and then eventually making sure that your primary endpoint is your lead endpoint, so that the FDA approves your drug product based on what that is. I think for these ultra rare conditions, at least when Dr. Marks was around, and from what we're seeing from the FDA right now, at least for the ultra rare conditions, they're not really focused in on is it your primary or secondary endpoint that you're seeing efficacy. Just show us efficacy. Show us that this disease is on a different trajectory than it was before. So, a really good mentor of mine was like, just get kids treated, you know?
Yeah.
And so we're looking at that in any way, shape, or form. Is that a compassionate use? Is that a phase 2B trial? Is that a phase 3? So that you're able to show at least in some way that the course of disease is different. And then once you get to that point, then you could decide, do you go for expedited approval because the efficacy is so strong? And if it's not strong enough, then you might need to do a phase 3 like us. And the reason for SPG 50 is not that we're not seeing efficacy. The problem is that how do you show a meaningful change on a slowly progressing disorder?
Yeah.
It's it's not like the children die in a year or two. It takes 10 years for someone to be paralyzed from the waist down. It takes 20 years for them to, slowly become paralyzed by their hands. And each child is acting differently. It's a broad spectrum of disorders. So that is our problem, and that's what we're working through. And we are seeing changes. It's just, is it different from the natural course of disease and what this patient should have been?
Yeah. How do you advocate trying to address that problem, either in SPG 50 specifically or more generally? What can people do if they have one of these cases where it's rare, and it's slower progressing? I mean, paraplegic by 10 and quadriplegic by 20 is a devastating outcome, but it's not death by 3, right? So I'd love to just have your explanation of how you can approach that.
Yeah, I think it's looking at the endpoints and making sure you have them all in there. For example, we didn't think cognition would be a big change, so we didn't really focus in on it, but it was one of the primary endpoints that really did showed a difference. Spasticity, surprisingly enough, that we thought would be a big, change, is not changing because people are not getting more spastic. At the
Mm-hmm.
same time, it's very hard to show the difference in spasticity because each patient. Cerebral palsy is where you get spasticity, right? And the biggest, most common disease. And spasticity in cerebral palsy, for the most part, doesn't change hour by hour, minute by minute. I can wake up in the morning with my son, put his AFOs on, and his feet could be completely loose, and he put his AFO on in a second. Or he could be upset in the morning, and it's really hard to do. It's
Yeah.
it's different than spasticity in the cerebral palsy patient. So it's really hard for us to determine, is there a change, because they can go into the hospital and they could be cold in the room or they could be unhappy and it could be showing more spasticity than what is really there. So it's very complicated. But what I would say is the FDA, who is the regulatory body that we care about, right, in the US, it seems to be very flexible. They
Yeah.
seem to care about us figuring out how do we move these forward. I would say more so than they were than the past. Like not, Mark's made a huge difference in where we are. Dr. Marks, I apologize. And I think that the FDA that is new today, they're people. These are individuals, are human beings that are there that want to help us make a change in these children. It's going to make sure that the risk ratio is there, right?
Yeah. So with that in mind, is the name of the game trying to assemble as multimodal, and clear of a picture from a data perspective, in both treated and non-treated patients, right? So you can, with some statistical rigor, look at a combination of things and say, we weren't able to perfectly predict that it'd be spasticity ahead of time. But if you look at these 3 or 4 things that all the clinicians agree matters and all the families agree matters to quality of life, then you can see that really clear picture
Yeah,
emerging.
and sometimes you don't see that. Like, how do you catch my child is more affectionate?
Yeah.
My child plays with his toys better. My child is able to climb in the bathtub still, or bathe himself, or excuse me, feed himself, or various other things that they normally wouldn't do,
Yeah.
and are not part of an endpoint like standing or crawling or the traditional ones.
How much does the FDA listen to that? If you were to collect that, how much do they care about patient or family quality of life type of structured data? Does it factor into the equation in any meaningful way?
Unfortunately, I'm not far enough down that path to tell you if they do or not,
Yeah.
because we're opening up a phase 3, we're doing other things. I think that, again, from my interactions with the FDA, I think that if you were to bring video interactions and patient testimonies, and I think they would very be open to listening to those items, but we still have to show some statistical difference, right? Like if you can show a statistical difference and have those patient stories, I think it could be, you'd have a case forward. And again, from from what we're seeing and from our submissions, the FDA is really trying to be flexible in moving this forward, at least from the phase 1, 2 perspective and going into the phase 3. We thought we had a really good endpoint. They came back and said, you really don't. And it wasn't like they were trying to be a roadblock. What they were doing was they were seeing what they were getting from other programs
Right.
and really guiding us to say you can't use, a GMFM88 as a whole. You're not going to get enough positive data from that. You need to choose a few really good endpoints and use them as your validation. They were guiding us towards what they're seeing in the field and helping us, from our expertise, because not everybody's an expert, trying to figure out what to do and how to move forward.
Yeah. You talked about the priority review vouchers earlier. I'd love if you could just explain that to people who aren't familiar with it, because it is a really clever and important way of breaking down this market failure that we have. And I am also curious. I think you're part of the FDA roundtable in June, and I know you stay close to the evolving sentiment legislation on this. Do you think are there whispers that it's coming back? Are there plans for it to come back? Because certainly from where I sit, I think it's incredibly important, and it would be a shame to have it go away.
Yeah, so the way I understand it, and I'm hoping I explain it correctly, is many years ago, they had this idea where large companies that made large commercialized drugs would take some of their funding and their R&D research and make drugs for small diseases, small disorders. So like, let's say a Roche would, or a Novo Nordic would make a small, pediatric condition drug, gene therapy, monoclonal antibody, small molecule, whatever it may be, and make a therapy for a small indication. And by doing so, when they went to the FDA for their next drug, they would shave 6 months off their application. So let's say they have, a drug that's going to make them lose weight, people lose weight, and it's worth $8 billion. You shave off 6 months, that's $4 billion more profits. Unfortunately, what happened was large companies to make a drug costs, $500 million. So it didn't really make sense for them to invest this much money into it. So the paradigm changed, I don't know exactly when, to instead of making drugs for small diseases, buying the priority review vouchers off of smaller companies, which then allowed them, the smaller companies, to use that money to pay back the R&D and all that other stuff and fund most of their new R&D, because
Yeah.
the vouchers were selling initially at $300 million. Then they came down to about 100 million. And now the vouchers, because they haven't been renewed, like you mentioned, are selling for around 150 million plus. Do I think they're going to get renewed? I personally think that it's a bipartisan, there's no bipartisan problem here.
Yeah.
Obama renewed it, Trump originally renewed it. And I think what happens is unfortunately, there's so many changes that are going through legislation right now that, that it just gets deprioritized every time it comes up. And so I think it's going to be a matter of time for enough people to make enough noise and for it to be brought up just enough for priorities to be seen for it to get renewed. I think that's the problem. It's not that people don't want it to be renewed. I think it's just, there's just too many changes in the US right now for it to be bubbling up to where it's supposed to be.
Yeah. And I'm sure it has a dramatic impact on companies like yours, right? Where the first thing an investor will do is they'll look at that uncertainty, right? And say, I can't necessarily count on this coming back. And so then they have to underwrite the whole rare disease pipeline differently than they might have otherwise, right?
Yeah, I mean, so there's that, and then there's also the funding for rare diseases. So, you used to have grants, you used to have, SBR grants, FDA grants, and those are less and more competitive now. And more places are now focusing in on larger indications, less rare. And then you have this peer review voucher that you could have used as a, deferral of payment process or sometime return on investment, and that's gone now.
Yeah.
So it basically leaves us in the rare disease community with literally no funding. And especially if more and more programs are being dropped, then where does it leave us? It leaves us, unfortunately, for patient and families to have to raise the money in order for us to get the treatment to patients.
Through all of this, you're a hopeful guy, an optimistic guy. It seems like you've pivoted your business as well to try to make the most of the challenging situation. Where do you see the light or the optimism? Is it in the science? Is it in the families? Is it in some combination of things?
On programs that we thought were dead, like for example, CLN7, an amazing mother from Texas treated 4 children. Unfortunately, her child was just a bit too severe to be treated. And she made the therapy. They treated 4 children. They showed efficacy in seizure activity, but no one could fund the rest of the program. So we've been helping the program on the side, trying to move it forward. But the reality was there was no funding. And then it took one family in Brazil to come around and say, you know what, we're going to do everything humanly possible to save our child and the other children. And they raised $3 million in like 4 months, 5 months. So a program that was completely dead got revitalized by a family. They funded the manufacturing, and now we're trying to figure out how do we pay for the treatment costs at the hospital. So I think, when I look at Glimmer of Hope, I see amazing families really stepping up. And then, for example, for CMT4J, we were stuck, and then we were able to move forward with some great news that will come out in the near future. But that's what's happening. It's amazing institutions, organizations, and families coming together to help bring these forward. What I see, unfortunately, I think we have another year and a half of hardship coming. I think until more and more programs, I think what we're doing is we're focusing on the bad things instead of the good things,
Yeah.
right? We focus in on 2 children that died in Sarepta's program, but we forgot about the 1,000 that were treated,
Right.
right? And I think until we start looking at how many patients have been treated in gene therapy compared to the losses, how many patients are doing better than they would have been before, looking at the positive rather than the negative, then I think things will turn around. I think as long as we continue moving forward, we continue making progress, more gene therapies get approved, we will come out of this, we'll come out of this stronger. And then what will happen is we'll have amazing technologies like lipid nanoparticles and gene editing or prime editing that are going to make us to get us to the point where in 10 years from now, a baby will be born. 7 days later, we'll have a genetic response back. We'll know that this child has SPG50 or some other God forbidden disease. And they'll plug into a computer. Someone in some facility will make the drug. And, on the 10th day, this child will be treated and never have this disorder.
Yeah.
I'm praying that we'll be there in 10 years. But it won't happen unless we take risks now. And I'm not saying that like just willy-nilly. Like we're not taking
Yeah.
risks
Informed risks, calculated risks. Yeah.
informed, calculated risks where safety is paramount. But when you don't know, you calculate your risk, you do everything you humanly can to mitigate that risk, and you treat the patients. But after treating 1,000 patients, when you treat a 53-year-old patient, because they're more, progress of the disease, these are the kind of things that are expected to happen. But, families want to take the risk, and we want to see how far we can go, right?
As we wrap here, how do you think people who listen to this podcast can best help you or help people in the rare disease therapeutics ecosystem more broadly?
Yeah, I would love people to help us, and if you can donate or if you're an investor to help us. But I would say if you could, more importantly, if there's a person, a family member, a neighbor, a cousin, a friend that has a child with rare disease, go and buy them coffee, take them out for a drink, just get them out of the house and listen to them, because it's really hard to have a child with a rare disease, a disabled child. If you see a family in your community that's struggling to raise money to do something like I did, which is a monumental task, go and do a lemonade stand. Go and have your kids do a lemonade stand in the front of the house, or do a gala or something for them. Help them move their program forward, because this is not easy. This is not meant for us.
Yeah.
It's not meant for a family to create. It's meant for a company to do so. And if they've really put themselves out there and been brave enough to do so, then you should try to help them.
Yeah. Thank you, Terry. You inspire me, and I know you inspire thousands of people every day to get up and try to make a difference here. So appreciate you taking the time to share your story with us, but also all the amazing work that you do for kids around the world.
Anytime. Honestly, like I think the impossible is to do nothing. I know that it's hard to say, and people are just living. Some people are just living through this disorder, barely living. But if you commit yourself and you say you're going to do this, it's possible. You just gotta just go all in. You know, you
Yeah.
have to go all in.
Yeah.
So
Amazing. Thank you.
awesome. Thank
And
you for your
thanks
time.
everybody as always for listening. We'll see you next time. Thanks as always for tuning into the Genetics Podcast. If you enjoyed today's conversation, the best way you can support the show is by sharing it with a friend or colleague who might find it interesting as well. We'd also really appreciate if you could subscribe, rate, and review us on Apple Podcasts, Spotify, or wherever you listen to podcasts. This helps other people discover the show when they're searching for content on genetics, precision medicine, or biotech. We always want to hear from you as well. If you have feedback or questions or want to be featured on the show, you can email us at podcast@sonogenetics.com. You can find me on LinkedIn, Patrick Short. You can find us as well, Sonogenetics, on LinkedIn, or reach out on Instagram at The Genetics Podcast. And finally, a special thank you to the team behind the show who makes this possible. Joy Ismail produces and manages the show. Jane Reddy Webster keeps us organized, and James Pierce from Selective Frequencies for his expert audio engineering. I'm Patrick Short, your host. Thanks again for listening, and we'll see you next time on the Genetics Podcast.